All right. So protein synthesis, pretty easy, okay? So first we need to recap what we know about DNA. Okay, so remember DNA is double-stranded. Hydrogen bonds hold those two strands together. It's made of nucleotides, which are the monomers, okay? And a nucleotide is made of a sugar, a phosphate, a nitrogen base, and those A, T, C, and G are the 4 nitrogen bases. Genes. We talked about this in the last unit. We're going to go a little bit deeper into it this time. Okay, genes are just a segment of DNA that code for a specific protein. Now, based on that video we watched, everything about you is made of protein, okay? Everything about you is made of protein. Each gene you have is made of protein. But remember how I told you, you have a whole bunch of DNA that doesn't do anything, okay? So we're going to be focusing on just those genes. This video is a great example of what genes are. How do you know they're a gene? Basically brings it down to a very fundamental level. It's a very good video.
Stated Clearly presents, what exactly is a gene? Each one of our cells contains 46 strands of DNA. A single strand is made of millions of particles called nucleotides, and these nucleotides come in 4 different types, which scientists have labeled A, C, T, and G. A gene is a special stretch of DNA, a sequence of A's, C's, T's, and G's that code for something. A gene contains information for a cell to read and use, but what exactly does that information do? You might have heard that there's a blue-eyed gene, a freckle gene, possibly even an anger gene, but single genes don't literally make things like eyeballs or freckles or temper tantrums. Genes make proteins. Those proteins then interact with each other and all sorts of chemicals inside the body to build things like eye pigments, freckles, and mood-altering hormones. A single strand of DNA contains thousands of genes or unique protein recipes. Humans have roughly 20,000 altogether. Some genes are small, only about 300 letters long. Others are well over 1 million. The length and sequence of a gene determine the size and shape of the protein it builds. The size and shape of the protein determine the function that protein will have inside the body. Hemoglobin, for example, is a protein structure found in red blood cells. Its unique shape and size allow it to capture oxygen molecules when blood flows near the lungs, and then release them later when blood flows near oxygen-starved tissues. Pepsin is a digestive protein. Its unique shape allows it to break down food inside your stomach so it can be absorbed by the body. Keratin is a structural protein. Its unique shape and size allow it to link together with other keratin proteins to form hard structures like fingernails, claws, and beaks. Different creatures have different genes, which is ultimately why their bodies look and function differently. But one of the many reasons scientists believe all life on Earth is related is that the basic DNA code, the language of A's, C's, T's, and G's, is pretty much the same for all living things. Many creatures even share some of the same genes. You might not be too surprised to learn that humans and chimps, which are closely related, share 96% of their genetic code. But what would you think a lowly fruit fly has in common with a beautiful swimsuit model? Surprisingly, about half of its genes. Because all creatures use DNA in pretty much the same way, genetic engineers have found that if they take a gene from, say, a bacteria cell and insert it into the DNA of an animal or a plant cell, that animal or plant cell will then read the new gene and produce the bacterial protein. Engineers have mixed and matched the genes of different organisms to produce many new creatures, including corn that is toxic to insects but supposedly safe for human consumption, tomatoes that last up to twice as long in the grocery store before going bad, and a new form of bacteria that produce the human protein insulin, which we then collect from these bacteria and give to people with diabetes who need extra insulin to survive. So just to sum things up a bit, what exactly is a gene? A gene is a special stretch of DNA, not the entire strand of DNA, just a segment that codes for something. Each gene is like a unique recipe, which usually tells a cell how to make a protein or a group of proteins. Different creatures have different genes, but all genes are written in the same basic DNA language of A's, C's, T's, and G's. I'm John Perry, and that's genes stated clearly.
Okay, so in that video, they show you some really good information and basics about genes, okay? Now, what was really cool about that is that kind of leads into, if you've heard about CRISPR, and how CRISPR is a gene editing machine. Well, the awesome thing is that, yes, we are capable of editing genes, okay? But that starts to walk a fine line on ethics, okay? We're not going to be ethical and worry about that stuff. I'm just kind of giving you some cool ideas. They can take the glow-in-the-dark gene from animals such as fireflies and put that gene into the embryo of a mouse or a cat. And guess what happens when that mouse or cat is born? Some of them glow in the dark. Maybe their eyes, maybe their feet, maybe their ears, maybe their whole body, okay? Because they got that gene. But on the same note, just because you have a gene does not mean you will see it or that it will be expressed. So on this slide, what I want you to focus on is that 3 nucleotides are called a codon, and that the order of these nucleotides or codons determine gene expression. Now, what gene expression means is that the gene can be seen in some capacity. For example, the hair you have on your head, I can see that it is a certain color and that it's curly, it's wavy, or it's straight, right? That is a gene that is being expressed and I can see it with my eyes. Now, do you have other genes that are being expressed that I can't see? Yes, but that's just an example. Just what you can see is a good example of genes that are being expressed, meaning this whole process of protein synthesis has been done to that specific segment of DNA or to that gene. My example for you is, if you have parents with different color eyes, whose eye color did you get? Some of you got your mom's, some of you got your dad's, but do you hold the ability to have either color? Yes, but your body did not do protein synthesis on both colors. It picked one color or the other, did protein synthesis, and now your eyes are that specific color, okay? My husband has brown eyes, I have blue. Our daughter has brown. But that doesn't mean she didn't get my blue-eyed gene, it's just not being expressed. Does that make sense? So we didn't do protein synthesis to her blue-eyed gene, we did it to her brown-eyed gene, because remember, she got one chromosome from mom and one chromosome from dad, and it picked which one it was going to do. Same happens for you if your parents have different hair color or hair type or eye color. You got one or the other. It's not that you don't have them both, it's just your body's only going to express one of them, okay?
What does it mean when neither, when the kid has neither one of the...
So if mom has blue and dad has brown and the kid has green, that means green exists on probably both sides, but they were not being expressed in either one of the parents, but they passed that gene on to their kid, and then they ended up with only a green option, so they got green. What? We're not going there. We're being real. We don't want to be negative. All right. So first step of actually getting these genes to be expressed is we have to do a process called transcription. This is essentially step one, okay? Transcription starts in the nucleus, because where's our DNA? In the nucleus. Now, our DNA is super important. We're never going to let it leave the nucleus. It's always going to stay there. And because it's never going to leave the nucleus, we have to make a specific version that can leave the nucleus, that we don't care if it gets beat up or messed up, that it can leave, and that's perfectly fine. We call it mRNA, which stands for messenger RNA, because they carry the message, okay? I have the answer key. I have all the originals here. Do I give out my originals to you guys? No, because then only one person would get it. And it would only be good one time. So what do I do instead? I make copies and I give you the copies. And then you lose a copy, guess what? I just hand you a new one. You break the copy, I hand you a new one. You rip the copy, I hand you a new one. But I never give you my original, because if you mess up the original, we got nothing to go back on, okay? So that's what's happening when we go from DNA to messenger RNA. So look at this picture of messenger RNA and tell me, what are two differences you see in that strand of nucleotides? One of y'all say one say the other. Okay, so U goes to A, all right. What letter is missing from the RNA? Not C, T. Okay, so we're getting rid of a nitrogen base, but all that is telling us, hey, this isn't DNA anymore. This is the copy. This is that messenger RNA. So now when you see strands of letters or nitrogen bases, you can go, that's DNA. Oh, that one, it's got a U. It must be RNA. Well, how do we make it? It's very, very easy. First, we got to remember what an enzyme is. You don't have to write this down. We just need to know. So they're proteins. They speed up chemical reactions. They're specific and they're reusable. Again, you don't need to write this down, but you do want to make sure you remember it, okay? All right. If y'all remember in DNA, when we made a new strand of DNA, we used DNA polymerase. Well, if we're going to make RNA, we're going to use a new enzyme. It's called RNA polymerase. You're not going to be tested on it. That's why it's not on your notes. I'm just telling you about it so you understand why that enzyme set was there as well, just to kind of Oh, okay, like it doesn't just magically happen. Something specific is doing it. Instead of DNA polymerase, it's RNA polymerase, okay? So RNA, it's got its own few characteristics. All right, so RNA is similar to DNA. It's made up of nucleotides, same thing, but now it's little sugar. Instead of being deoxyribose, it's actually just ribose. Instead of being double-stranded, it's single-stranded. And we're gonna get rid of thymine, and instead we're gonna have uracil. So now our A is gonna pair with a U instead of a T. So here's our 4 bases, A, U, C, G. And now this is the nucleic acid that can actually leave the nucleus, because remember, it's just partial information. It's just a disposable copy, and it's just a section. It's not all of it. It's just that section of DNA. It's not all of it, okay? So how do we do transcription? Well, let me show you. So in your notes, you go to the next page where you have all these graphics. At the top, there are 2 strands. We're going to do those strands together to make sure we got it, okay? So first we need to make a little nitrogen-based key. So DNA are A, T, C, G. RNA code is U, A. So this is what we're going to be using, okay? But sometimes we forget that T still goes to A, but A is going to change and go to U. So on your paper, make sure that you're not writing this on the amino acid line. Keep it on the mRNA line directly below. So A turns into a U. Continue on practicing to see if you can finish this out. And now that you've had a chance to kind of work through it, check your answers. How did you do? Mm-hmm. Okay. And if you notice, I didn't put any T's down here. You should be T free. Okay.